Immunology and immunity in health and diseases. Immune response against parasitic infections. This particular study note is developed by EPG Pathshala team in HID. So I would like to express my sincere gratitude to development team. Thank you so much. Here we are going to learn about immunity in health and diseases. Immune response against parasitic infections in particular. Parasite host interaction, immune response, infection, and immune evasion. Our immune system is the sum total of all the strategies that have evolved to protect our body against parasites or altered cells. Once a parasite is recognized, the immune system recruits a dynamic network of various kinds of cells and molecules to mount an appropriate response called an effector response to eliminate or neutralize the organism. The body resists an invading parasite with full force. Subsequent exposure to the same parasite generates an appropriate memory response involving a quicker and higher level immune response to eliminate the parasite and prevent infection. However, in some cases, the immune response may be weak, or the parasite is able to overcome host resistive forces and establish itself inside the host. They may cause chronic, life-threatening infections, diseases to the host. Thus, the establishment of a parasite depends on the outcome of the defensive forces employed by the host against the parasite and the evasive strategies of the parasite. This module presents a broad overview of the immunological aspects of parasite invasion in a simple yet comprehensive manner. The first half highlights the role of three-tier defense system of our body that works against the parasite attack. Then both non-specific and specific responses are explained, focusing on how the protozoan parasites are countered and how helminth worms are expelled out. The second half discusses the strategies employed by the invading parasites to avoid the host immune response. Various methods that thwart the host immune responses are described. The term parasite is used to denote an organism that is dependent on another organism, the host, for its survival. Parasites may live on or the body of their host and derive nourishment and habitat from it. The host in a host-parasite reaction is a habitat for the parasite to colonize and the source is to be exploited. The parasite is usually smaller than the host and may infect an invertebrate or vertebrate, but it's specific for it. Parasites have a full range of symbiotic interaction with their animal hosts. Generally speaking, parasites include the unicellular eukaryotes, the protozoa, and the multicellular eukaryotes, the helminths. So, protozoan parasites are microscopic protists that may grow within or outside the cells, often multiplying themselves within their host. They are responsible for causing several life-threatening diseases in humans like malaria, Chagas disease, African sleeping sickness, leishmaniasis, then amoebiasis, and toxoplasmosis. Helminths, commonly called worms, include the nematodes, roundworms, the trematodes or flukes, and the cystoids, tapeworms. They do not ordinarily multiply inside the host and remain extracellular, inhabiting the host tissues or bloodstream. These are big worm-like parasites, thereby displaying more number of antigens on their body surface. All these factors make them more accessible to the immune system. However, the level of immune response generated against helminths is very, very low, owing to their low copy number and various other strategies they employ. So, pathogenicity may be a manifestation of the host parasite infections. It is an outcome of various factors like ability of the parasite to invade the host, to multiply in the host's body, to avoid host defenses to establish itself on the host, and number of infectious organisms entering the host body. Both protozoan and helminth parasites causes a large repertoire of infectious diseases. These infectious diseases remain a wide major cause of mortality and morbidity for millions of people across the world, some of which are listed. We could see amoebic dysentery, giardiasis or diarrhea, amoebic dysentery, amoebiasis, entamoeba histolytica, fecal oral route, sexual transmission, vectors such as fleas, cockroaches, etc., large intestine, site of parasitism. Giardiasis or diarrhea, Giardia intestinalis, food or water contaminated with infected cysts or sexual act. Duodenum and jejunum of small intestine. Malaria, Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, female Anopheles mosquito. Hepatocytes, RBCs. Cutaneous leishmaniasis, Kala-Azar fever, Leishmania major, sandfly, macrophages. Then visceral leishmaniasis, Leishmania donovani, sandfly. Then trypanosomiasis, African sleeping sickness, Trypanosoma brucei, Trypanosoma gambiense, Trypanosoma rhodesiense. Tsetse fly, bloodstream. Trypanosomiasis, Chagas disease, American sleeping sickness. Trypanosoma cruzi, tritomid bug, blood parasite. Basil. Toxoplasmosis, Toxoplasma gondii, infected stages in cat feces, undercooked meat, many cell types. Then trichomoniasis, vaginitis or leucorrhea. Trichomonas vaginalis, vagina female, sexual act. Trichomoniasis. Then blandidiasis, Blandidium dysentery, Bacterium coli, intestine cyst with contaminated food and water, large intestine, colon. Helminths, small intestine, trichuriasis, colon and cecum. Ankylostomiasis, small intestine, enterobiasis, cecum and colon and appendix. Pinworm. Then tissue nematodes. Trichionellosis. Then lymphatic filariasis, onchocerciasis, then loesis, eyeworm. Trematodes, schistosomiasis, portal and mesenteric veins, bloodstream. Cystodes, Teniasis, Teniasolium, pig tapeworm. Cysticercosis, larvae of the Teniasolium. Hydatid disease, small intestine, dog tapeworm or hydatid worm. Host defenses against parasites. As the parasite attacked the host, the two came into a close dynamic interaction. While the parasite tries to establish itself in or on the bodies of host, the host mounts a series of immune defense actions against the parasites. The outcome of these two actions depends on the degree to which they are effective, or the stronger wins over easily. Typically, the host defense mechanisms are divided into two major subdivisions: innate or non-specific immune system and adaptive acquired or specific immune system. First line of defense, innate defenses, barriers at the body surface, skin, body secretions, mucous membranes, cuff reflexes. Second line of defense, internal innate defenses, inflammation, phagocytosis, complement system. Third line, acquired adaptive response, B lymphocytes, antibodies, and T lymphocytes, non-specific and specific. Innate defenses of the body are non-specific. They respond to any type of parasites with similar response. These defense mechanisms provide first-hand protection against invasion and are present in the body even without any prior exposure to that pathogen. Innate defenses include anatomical and structural barriers, or the first line of defense of the host's body. These are external defensive forces that act at the body surface level. These include epithelial surface of skin, except at places of cut or abrasion, body secretions, tear, saliva, sweat, and secretions of the gastrointestinal and vaginal tracts, which flush out invading parasites, movement due to cilia and gut peristalsis, mucous membrane lining the respiratory gastrointestinal tract, cuff reflexes. Parasites tend to be more specific when it comes to the kind of host they will infect. This host specificity is governed by the parasite's preferences, age, sex, and gender of the host, and also by the broad molecular patterns involved in host cell recognition. Parasite-associated molecular patterns, PAMPs, are distinct evolutionary conserved structures present on the parasite surface. They recognize the bind to pattern recognition receptors, PRRs, a family of toll-like receptors on host macrophages and dendritic cells. Specificity differences can also result from subtle variations in genetic determinants of the host. It has been found that individuals heterozygous for the sickle cell possessing a distinct form of hemoglobin are more resistant to Plasmodium. Similarly, absence of Duffy blood group antigens (FY) makes an individual resistant to malaria, as seen in many parts of Africa. This happens because Duffy antigens act as the membrane receptor for Plasmodium vivax. Also, sheep with hemoglobin A mount a more effective self-cure immune response against Haemococcus contortus and Ostertaggia circumcincta and those with HBV. Another parasite-derived factor is that the presence of one parasite may affect the growth of the another at the same host. For example, the presence of adult worms in the host body often delays the establishment and development of larval stages of the same species. This phenomenon is concomitant immunity has been seen in the case of Manzoni Echinococcus granulosus, that is concomitant immunity. If a parasite breaches the external anatomical barriers and invades the host tissues or enters the bloodstream, the internal innate defense of the body, second line of defense, come into play. These include inflammation. The PAMP-PRR recognition activates the macrophages and dendritic cells into release cytokines IL-2, IFN gamma, and TNF alpha, triggering intracellular signaling cascade that help in generating innate immune response. The cytokines also harmonize the influx of inflammatory cells, phagocytes, and neutrophils to the site of parasite infection. These effects include vasodilation, increased capillary permeability, accompanied by swelling, pain, redness in the infected areas. The amount of inflammation can be correlated with the extent of infection or parasite load in the host. Chagas disease caused by this organism of T. cruzi is characterized by inflammation or lesions initiated in the colon, which under chronic conditions spreads over to heart, kidney, and other organs. Rupture of infected red blood cells in Plasmodium infection releases uric acid, which contributes to the massive inflammation, triggering the onset of cerebral malaria. Helminth infection also causes local inflammation in the gut epithelium. Female schistosomes lay eggs on the epithelial lining of the venous capillary walls, causing inflammation in liver or lungs. Then phagocytosis. Circulating polymorphonuclear neutrophils or PMNs, monocytes and macrophages play a role in phagocytosis, the act of recognizing and ingesting parasites for intracellular killing as part of innate defense mechanism of the host body. The process of phagocytosis by an activated phagocyte that has recognized and internalized a parasite for removal from the body. Phagocytic cells, neutrophils, and macrophages engulf the parasite and respond with the increased granular enzyme that digests intracellular parasites. Phagocytes also display a mechanism of respiratory burst, the production of reactive nitrogen species such as NO, RNS, reactive nitrogen species, NO by macrophages, and receive oxygen species, reactive oxygen species such as H2O2 by neutrophils, crucial for the killing of intracellular parasites. However, this reactive species also cause some tissue damage, which ultimately leads to increased vascular permeability and organ injury. Parasites undergo internalization and processing inside APCs and are generated on MHC2. Macrophages can be activated by innate mechanism of IFN gamma production by NK cells, as well as the adaptive process mediated by activation of Th1 and production of IFN gamma. Complement activation. This involves a cascade of complement proteins, factors, that bring about the lysis of invading microorganisms. Complement factors are a family of membrane associated glycoproteins, proteolytic enzymes that play three major roles like opsonization. C3b acts as the major opsonizing agent, then inflammation, complement breakdown products such as chemoattractants, attracting macrophages and neutrophils. Then membrane lysis, complement proteins form from a membrane attack complex MAC that ruptures parasite cell membrane and enhance their phagocytic killing. Thus, when a pathogen enters the body, the host initially attempts to eliminate it by innate mechanisms. These mechanisms are immediate and try to control the infection while the adaptive response is being developed. However, they neither provide long-lasting immunity or nor memory. Also, if the pathogen has previously infected the animal, adaptive acquired immunity operates immediately to expel the returning invader. In fact, an immune response, including both innate and adaptive responses, is elicited by parasitic infections. Specific adaptive acquired immune response towards parasitic infections. Two types of adaptive immune response follow a parasitic infection: humoral and cell-mediated. Professional antigen-presenting cells, APCs, macrophages, B lymphocytes, and dendritic cells internalize the parasite, process, and present their antigens on antigen presentation machinery. Major histocompatibility complex, class 2. Then MHC complex activated CD4 plus TH cells, which may further differentiate as TH1 or TH2 subtype. These subtypes differ in their mechanism of action, cytokine produced, and the kind of parasites they are effective against. However, a general proposition is that a TH1 mediated cellular DM response will target the intracellular parasites growing within the macrophages, while a TH2 mediated humoral or the antibody mediated immune response will be developed against extracellular parasites or stages living in body, blood, body fluids or gut, blood-borne sporozoite and merozoite stages of Plasmodium, Trypanosoma brucei larvae. Expulsion of helminth worms inhabiting the gut or tissues requires a well-coordinated, tricky operation of both the humoral and CMI response. A schematic diagram is shown, which we will be reviewing later. TH cells carry out cell-mediated immune response. TH1 cells produce a number of cytokines, IFN gamma, TNF beta, IL2, IL12 as part of CMI response in killing cells harboring obligate intracellular protozoan parasites, macrophages in infection with L mode major, T. cruzi and gonads and Eimeria sp, and the liver and erythrocyte stages of Plasmodium. IFN gamma has many effects, including activation of macrophages with high production of nitrous oxide that is lethal to intracellular parasites. IL2 stimulates the maturation of TC cells, B cells, and enhances the cytotoxicity of NK cells. IL12 induces the secretion of IFN gamma. Then TH2 cells bring about a strong humoral response. A strong humoral response requires the inputs of TH2 type cells in destruction of helminth parasites. The production of IL4 by NK cells, gamma delta cells, mast cells, basophils, and eosinophils induce the self-differentiation of both TH cells into TH2 type. TH2 cells also secrete IL5, IL6, IL10, and IL13. These cytokines help B cells to undergo class switching and produce IgE, IgG1, and IgA for activation of particular B cells to release antigen-specific antibodies. Antibodies act as opsonizing agents, promote phagocytosis, and also mediate complement-mediated lysis of the infected cell. High levels of antibodies found in the blood of infected patients than in non-infected ones have been correlated at resistance of the host against infection. Some B cells form antibody-forming cells, while a few form B memory cells. T-cell mediated cytotoxicity is carried out by CD8 plus TC cells or through a secretion of cytokine by CD4 plus TH cells, which activate and mature B cells, natural killer cells, and macrophages to destroy intracellular parasites. Macrophage activation ennosynthesis. CD4 plus T cells, TH1, TH2, CD8 plus T cells, CTL, kill infected cells. It is interesting to note that CH1 and CH2 responses are antagonists to each other. IFN gamma negatively regulates TH2 response, and IL4 and IL10 suppresses TH1 response. This helps to maintain a state of homeostasis in the immune system, which is very essential. It has been seen in some cases that inappropriate or extensively simulated immune response itself causes immune damage to the host. like Chagas disease. Much immunopathologic symptoms are caused in the host by CD4 plus CD8 plus cells. Similarly, overproduction of Th1 cytokines, IFN gamma and TNF alpha and NO have been found responsible for the neurological lesions and obstructed neurotransmission respectively in patients of cerebral malaria. Immunity against protozoan parasites. Generation of the appropriate Th-type immune response is critical in combating the protozoan infection. The chief executive powers of immune response against intracellular protozoan parasites are B cells with their antibodies, T cells and their mediators, cytokines, parasitic harboring macrophages, antigen oxide mediated killing and NK cells. Cellular immunity, the protozoan of the polarization. The polarization of Th cells towards Th1 type response is critical in immune response towards obligate intracellular protozoan parasites. As discussed in the section genetic determinants, the elimination of L major and resistant forms is significant. Delay depend on the development of Th1 cells which release IFN gamma and activate macrophages to kill intracellular mastigotes. The importance of Th1 in protection from T. gonadi and T. cruzi is also proven experimentally. Through Chagas disease caused by T. cruzi cannot be controlled completely by Th1 response. In Plasmodium infections, both Th1 and Th2 type CD4 plus T cells are involved in protective immunity. Though the relative contribution of these subsets changed during the course of infection, Th1 cells act primarily during the acute phase and Th2 cells are predominant during later phase of infection. CD8 plus T cells carry out killing of the intracellular stages of Plasmodium sporozoites inside hepatocytes and merozoites inside RBC, possibly by reducing chemocytokines, IFN gamma, TNF alpha, which produce the production of NO inside these infected cells. Humoral immune response. Next is about humoral immune response. A strong humoral immune response undergoes against the coat of variant surface glycoprotein VSJ antigens of African trypanosomes. This eliminates most parasites by antibody opsonization, phagocytosis, and complement-mediated lysis. However, some approximately 1% parasites which have a variant VSJ survive and further multiply in the host blood, causing another round of parasitemia. These successive waves of parasitemia keep occurring, including the steps: development of humoral immunity, killing of existing strains, escape of variant parasites, proliferation of new variant strain. Humoral immune response. A strong humoral antibody response developed against the coat of variant surface glycoprotein of African trypanosoma. Then this eliminates most parasites by antibodies opsonization, phagocytosis, and complement-mediated lysis. In Plasmodium infection, the sporozoites remain in host bloodstream for about 30 minutes before entering the hepatocytes. This time is insufficient to generate a good antibody response, though some antibodies are generated which prevent infection of liver cells to some extent. Merozoites, when released by erythrocytes, also generate a humoral response before infecting fresh erythrocytes. Antibodies block the capacity of sporozoites and merozoites to invade new cells. The tachyzoites of T. gonadi also trigger some Th2 mediated B cell response during their brief transit between the rupture of infected cells and infection of new cells. In general, infections caused by Trichomonas vaginalis, allergic reaction caused by local production of IgE and IgG tend to vascular permeability, promoting immobilization and killing of the parasite. Immobilization and killing of parasite. I just got messed up. The importance of antibodies and immunity against protozoan parasites can be assessed from the fact that B cell mu-MT deficient experimental mice could not eliminate Plasmodium, rather develop a chronic relapsing infection with no memory response. Antibodies to each variant, weeks of infection. Role of NK cells. NK cells seem to play a major protective role in destroying host cells harboring the parasite. Activated NK cells primarily function in the lysis of protozoan infected cells and production of cytokines IFN gamma and TNF alpha that stimulate their other immune affected cells like macrophages. So chemokines produced by APCs, IL-12, TNF alpha, IL-1 beta, IL-2 active NK cells, then IL-10, TGF beta, and to some extent IL-4 inhibit NK cell activity. The role of IL-10 in regulating NK cell activity can be judged from the fact that mice strains deficit in IL-10 rapidly succumb to infection with an virulent strain owing to amplified NK cell activity and overproduction of IFN alpha, IFN gamma, TNF alpha, and IL-12. Also, monoclonal antibodies administered against IL-12, IL-10 and TGF beta also known to cause enhanced NK cell associated IFN gamma production in infected mice. Role of macrophages. Stimulated by IFN gamma produced by Th1 cells or NK cells, macrophages carry out intracellular killing of the engulfed parasites like sporozoite stages of Plasmodium, in liver macrophages, Kupffer cells, some other Leishmania. Macrophages also destroy other infected cells like RBC, rhozoid stages of Plasmodium by direct cytotoxicity. Role of nitric oxide. Infected macrophages are induced to increase the production of reactive nitrogen species RNS such as nitric oxide. Nitric oxide mediated killing has been seen effective against intracellular parasites like Leishmania, T. gonadi, IFN gamma, INF gamma and TNF alpha activate macrophages to generate NO, while TGF beta and IL-10 inhibit NO production. Genetic determination of host immune response against parasites. The type of immune response and the severity of the infection a protozoan can cause depend partly on the host's genetic determinants. Experimental mice infected with Leishmania major have reacted as genetically resistant or susceptible to the disease. The resistant strains are characterized by development of Th1 type response and production of IFN alpha by IFN gamma by parasite harboring macrophages and do not show any clinical symptoms of the disease, while the susceptible ones show a Th2 type immune response with raised IL-4 levels that display a progressive and ultimately lethal form of disease. Similarly, different strains of mice experimentally infected with T. spiralis and interstitial nematode show disparity in response. While one strain is able to expel worms successfully, the mediating a Th1 response showing high IFN gamma and IgG2A antibody levels, the other strain remains susceptible by demonstrating a Th2 response. High IL-4, IgG1 antibody but low IFN gamma levels. Researchers have also demonstrated the protective use of Th2 response and IL-5 production in expelling the intestinal nematode Trichuromyuriasis in one strain of mice, while another strain demonstrating Th1 response and IFN gamma production remained susceptible and developed a latent form of infection. Next major topic, innate response against helminths. Being large in size, helminths display a number of antigens, making themselves more antigenic. But their large physical size and cuticle covering may pose a challenge to the host immune system. These large complex worms attach firmly to the gut wall, feeding enormously on gut tissues or blood. Also, their thick cuticle cannot be penetrated by MAC assembly of complement system or by the perforin chemical factors released by NK cells. Thus, the host's immune system has a challenging role to play, which it executes via a strong Th2 type response. Humoral response. The best initiator of host immune response is released continuously by the worm itself. The secretory proteins or ES proteins that acts as antigen. This ES protein of the helminth across the mucosal barrier of the gut lining to be captured by APC, particularly macrophages within the Peyer's patches or mesenteric lymph node, paracortex and follicle. APC process and present these antigens to T cells which differentiate as Th2 cells. Stimulated Th2 cells produce cytokines IL4, IL5, IL9, IL10, 13 and help B cells to undergo class switching and produce antibodies IgE and some IgG and IgA. These antibodies probably cross over to the intestinal lumen to cause direct damage by binding to the parasitic surface. So antibodies may inhibit enzymes released by the parasite, thereby preventing its ability to attach host cells and penetrate tissues. Activation of Th2 cells, release of cytokines, helmin the ES antigens cross over through the lumen wall. ES antigen are captured by macrophages, activated by Th2 cells, release of cytokines, activation of B cells, release of antibodies. Antibodies cross over into the gut. Inflammation occurs, recruitment of eosinophils, mast cell degranulation. IgE binds to the Fc epsilon receptor on the surface of mast cells lining of the intestine. Cross linking of this IgE molecule by more ES antigens cause mast cell degranulation, releasing free radicals, prostaglandins, leukotrienes, proteolytic enzymes including mucosal mast cell protease, MMCP1, hydrolases and vasodilator histamine. So histamine causes vascular permeability and dilation of the intestinal wall. An acute inflammatory response pursues, characterized by infiltration of eosinophils to the site. Inflammation changes the structure and physiology of the intestine at the site of attachment of the parasite, making it difficult for the parasite to cling on. Eosinophils also bear IgE receptors and when triggered, release their granule products toxic to helminth parasites. The granules are rich in nitric oxide, lysosomal proteases, lytic enzymes and major basic protein MBP, all of which damage the worm cuticle. Eosinophils also contain cationic proteins and ribonucleases that act as neurotoxins, thereby killing the worm. This yields to type 1 hypersensitivity response characterized by eosinophilia, high IgE levels, edema, vascular dilation, probably involved for expulsion of gut-inhibiting worm parasites only. This results in detachment of the worm from the intestinal wall. IL9 and IL5 stimulate mast cell degranulation. IL5 and IL13 trigger eosinophilia. Further, the infiltrating T lymphocytes secrete cytokines IL3 and IL13, which cause proliferation and differentiation of intestinal goblet cells, discharging more mucus into the intestinal lumen. Mucus forms a densely glycosylated viscoelastic gel-like coating around the detached worm and facilitates its expulsion from the intestine, aided by gut peristalsis induced by other mast cell mediators like IL4. This is called the weep and sheep response of the intestine. The self-cure phenomenon of the host against a worm parasite, as has been studied against T. spiralis, S. manzoni, and others in swine. It is persistent knowledge that the protective energy employed for expulsion varies with the parasite, as has been found that different Th2 type cytokines may be produced against immunologically unrelated helminth parasites. Acute phase of schistosomiasis features secretion of IL4, IL5 and IL13. Raised levels of IgA have been found associated with this infection in sheep. Secreted IgM was essential for timely expulsion of Brugia third stage larva IL3. Different stages of worm T. spiralis display different surface antigens had to induce different classes of cytokines. Also, the type of reactions acting against a parasite depend upon the location of the parasite within the host body. When the parasite has invaded through the intestinal wall and reached tissue spaces, a different process of expulsion is seen to mimic the state of phagocytosis. As seen for cystosomes, IgE and IgG antibodies produced in response to worm ES antigens coat the parasite and also begin to neutralize any ES antigen further produced by the parasite. This bound antibodies mediate a direct recruitment of granulocytes, particularly eosinophils and mast cells, into the parasitic body wall through the Fc receptor mediated bonding. The cells degranulate locally and pour their granule mediated into parasitic coat, damaging the parasitic body in an ADCC type manner. Cellular response through Th2 type immune response is a universal feature of worm expulsion. characterized by elevated IgE levels and eosinophilia and host in some cases. Th1 type response may also offer some protection. CD8 plus T cells or CTLs may be activated against helminths, deeply invading the intestinal mucosa or undertaking tissue migration. Infected animals may show production of cytokines characteristic of Th1 response or delayed hypersensitivity DTH. CMI play a large role in suppression of E. granulosus larva growth and also regulate pro-inflammatory Th1 responses. Teniasis in mice models also displayed production of IL-2 and IL-5 gamma, but not IL-4. Evasion of host defenses by the parasite. The host and the parasite are in a dynamic equilibrium, the outcome of which depends on the balance between the parasite virulence and host defenses. Although hosts employ a number of protective strategies to evade the parasites, these strategies are not 100% effective. The parasites may successfully invade the host by virtue of mechanisms that allow them to evade host immune systems. With time, the antigenicity of the parasite may decrease, allowing them to survive for a long period of time inside the host. In addition, the occurrence and severity of parasitic infection varies greatly in different hosts. It indicates that in most cases, the immune system is unable to provide complete sterilization, though it does not allow large-scale multiplication of parasites or diseases. Actually, a successful parasite is one that survives in the host for its entire lifespan, even without causing disease, unless its balance is lost by death or death of the host. Avoidance of innate host defense. The parasites evade host immune strategies by the following means. Protective covering. Parasites may develop a protective covering of tegment that may be resistant to antibodies and complement lysis or protective cyst that renders the parasitic hypoallergenic. The tough cuticle of adult roundworms also protects them from enzyme degradation. Adoption of intracellular lifestyle. Parasites may be sequestrated in a particular anatomical position so that it may be difficult to access. Getting into the host cells allows the protozoan parasites to escape host-specific immune defenses like hematocyte and erythrocyte stages of Plasmodium and macrophage stages of Leishmania sp. Large size of helminth parasites. One of the primary reasons that many helminths retain impervious by the body's immune attack is simply their large size and mobility. Several centimeters long, while tapeworms may exceed 1 meter in length. Oh my god. Taenia 4 meter long. Adding to that, fast mobility allows these parasites to escape inflammatory attack. Minimal exposure to host immune system. The Plasmodium falciparum protein PFEMPI is expressed on the surface of infected erythrocytes, leading to rosette formation. Parasites now spread to new cells with minimal exposure to host immune system. Avoidance of complement-mediated damage. By producing anti-complement factors, larval cystodes produce anti-complement factors which protect their outer layers from lytic attack and prolong their survival. Resistant to MAC forming and inserting. Leishmania develop a long extended surface lipophosphogycan LPG that allows the initial binding of complement proteins but avoids MAC insertion into its membrane. The VSJ proteins of T. cruzi prevent assembly of the complement cascade. Secretion of diversionary structures, decoy cytokine receptors. Attenuating the activity of the alternate complement pathway. Phagosome formation, prevention, resistance to phagocytosis, prevent maturation of phagosome, inhibit phagosome fusion, escape from phagosomes of the phagocytes into the cytosol, escaping adaptive host defenses, antigen masking, antigen shedding, antigen mimicry, molecular mimicry and host immunosuppression, protein inhibitors, prostaglandins, antigenetic drift,